Welcome to the Global Addiction Therapeutics Innovation Day.
Hello, everyone. My name is Mark Thomas. I'm proud to be chairing this session with Mark Potenza. I'm a professor of neuroscience at the University of Minnesota and director of the medical discovery team at the University of Minnesota Medical School. So today I'm proud to be, our group is proud to be a sponsor of this inaugural innovation session, and thank you for the organizers for putting this together. It's my pleasure to introduce the next speaker, Dr. Marco Pravetoni. I've known him for quite a while. He had hair like this, I think, when I first met him. I was on his dissertation committee. I can't take any credit for the success he's had since then. It's my pleasure to introduce Dr. Pravetoni from the University of Washington, who will be talking to us about his innovations in immunotherapy for addiction.
Thank you, Mark, for the kind introduction, and thank you everybody for being here, and thank you to the organizer for inviting me here. This is the first time at ISAM. I'm very excited to be here. Also, the first time in Hamburg, very nice. Rains as much as Seattle, so I feel at home. And I'm sorry, louder. Oh, okay, all right. So, well, I will start. And can you hear me now? It's better. No. Sorry. Yeah. Okay. All right. Okay. Yeah. So today I'm going to show some of our work in the space of developing immunotherapeutics to treat and prevent opioid use disorders and overdose. For disclosure, I filed patents in this space. For this technology, I'm the founder and scientific officer of Contract Therapeutics. Our work focused mainly on development of vaccines and monoclonal antibodies to either treat or prevent opioid use disorders and overdose through either active or passive immunization. Subjects are provided with polyclonal or monoclonal antibodies that will actually bind the target opioid, prevent the entry of the drug to the brain, reduce overall free drug, and thereby blunting or reducing drug effects related to either reward or lapse or toxicity such as respiratory depression and bradycardia. And we have demonstrated some of these effects in various rodent as well as large animal models and now in humans. I'm going to give you some examples of this technology specific to vaccines, both either therapeutic or prophylactic. So this is our pipeline. We go from early discovery to phase 1 clinical trials. Our most advanced candidate is an oxycodone vaccine that also target hydrocodone oxymorphone. This is a phase 1 clinical trial, a double-blind placebo control that is currently ongoing in the United States. And then we are advancing to IND and FDA approval vaccines for either heroin and metabolites, fentanyl and analogs. And for these programs, we are essentially completing all the IND-enabling activities, including manufacturing, GLP tox, and so forth. And then we have our other programs that are bringing forward vaccines targeting carfentanil, for instance, or employing next-generation formulation or technologies to advance this particular approach. This is our platform. This is how you build essentially a vaccine against an opioid, but any drug or small molecule for that matter. There is the drug-based aptin that essentially is focusing the immune response to the particular drug of choice. This is oxycodone, for instance. The small molecule is attached through a linker and conjugation chemistry to a carrier, which is typically a protein, could also be a particle and so forth, to actually have the immune system to recognize the small molecules. And then tailoring to indication, vaccination strategies could include monovalent or multivalent, depending upon how many targets you are interested to hit. And then use of immunomodulators or formulation to further improve the immune response. And the overall goal is to induce antibodies that are selective, effective, and long-lasting for the target drug. The first step is to create aptin libraries. We develop dozens of compounds that resemble the target drug. Here I'm just showing you some example with fentanyl and some of the fentanyl analogs. The goal is to elicit antibodies that will recognize these aptins, but also cross-react with the target drug. And also maintain selectivities for avoid binding useful drugs like naloxone, methadone, and so forth. This is just an example of how they work. These are rats that are vaccinated against fentanyl with different vaccine formulation, and then they're either challenged through either acute or chronic models and dose ranges. And so here we are showing how, compared to control, like vaccines would prevent fentanyl-induced antinociception, respiratory depression, and bradycardia. If you're looking at the center panel, panel B, if you look at a rat that is down at 50% oxygen saturation, the control, the treated animals actually do not show these deficits. And we have shown like these effects in various animal models against like different fentanyl and fentanyl analogs challenge models. As far as more chronic exposure, we employ self-administration studies. These are animals that first acquire and then maintain self-administration IV of fentanyl, and then we start the immunization protocol. And after completing the immunization protocol, the animals will actually discontinue or like decrease compared to baseline use of fentanyl. And when challenged with those responses, the control animal like compensate like to adjust for a schedule while instead, or a dose, while instead the vaccinated animals do not. And actually quite surprisingly, like even when animals are after extinction are re-exposed to drug, like vaccines seem to be affecting reacquisition of drug intake, which perhaps could help in situations of relapse and reacquisition of drug-seeking behavior. As far as selectivities, these vaccines are very selective. We always screen them like for the target opioids, but also for off-target. So things like endogenous opioid interference, or things like drugs used in medication of opioid use disorders like methadone, naltrexone, like buprenorphine and all. There is no interference there. And also other medication used in critical care, for instance, like anesthetics, there is no interference there. Formulation, you can really be creative with formulations. Like we have been working for many years with different adjuvants. Lately we've been focusing on toll-like receptor 7 and 8, importantly because like TLR8 is expressed like in humans, but not in certain animal models. And here, like pretty much we always kind of run animals through the same battery where we are looking at various effects of the vaccine. And by adding TLR7 and 8 to an established like fentanyl vaccine, we are seeing that A, we have improved titers against fentanyl. B and C, we are showing that respiratory depression is more counteracted with this particular formulation. And finally, that like the effects of the vaccine are due to reducing drug concentration to the brain. And this particular formulation, we are now completing the manufacturing and the GLP toxicology studies, and hopefully in a few months we're going to file an IND with the FDA to actually initiate like phase 1 clinical trials. As far as like today, like all the other panelists also highlight the problem of like polydrug use scenarios. We are trying like to mix and match these vaccines to see if they can actually address polydrug use scenario. This is one strategy, making multivalent vaccines by essentially combining individual vaccines. In this particular case, we are looking at a pentavalent vaccine that was given as a shot. So a single shot would protect against like heroin, oxy, fentanyl, carfentanil, methamphetamine, and their metabolites or close analogs. And we're showing this by challenging animals with bolus challenges where like you can put 5 drugs all at once and then just inject it in the rat. And what we are seeing the pharmacology behavior is that the vaccine prevents the effects or reduces the effects of like these bolus cocktail doses. And when we are looking at the pharmacokinetics, we are seeing that like this protection is broad, but yet still selective for the target drug. And so this is one approach like to protect against polydrug use scenarios. Another approach like to kind of come up with like a single vaccine product that perhaps like instead of combining multiple vaccines, in particular case like with medicinal chemistry approaches, we had like aptans that can display like different structure. Here in this particular case is fentanyl and oxycodone. And with one single vaccine, we can actually cover both drug classes or categories, if you will, still maintaining selectivity. Obviously then when like you are scaling up, like some of the consideration to be made, like so it's a lot easier to make individual vaccines and then combine them than making or characterizing like a vaccine formulation like this one. So there is always like pros and cons, and depending upon what that needs to be advanced through the manufacturing process and what's feasible to be advanced like to the clinic. As far as like understanding how these vaccines go, when I start like I guess working with vaccines was the time where people were doing clinical trials of nicotine and cocaine vaccines, which essentially show that only top responders would actually show efficacy against like the target drug. And so for the initial part of my career, I essentially focused on understanding why and what if we can predict like a high and a low responder so that we can avoid some of the mistakes of the past. And so we employ like a suite of like assays and testing. So we go from in vitro in silico in cell lines or using like human blood and AI and machine learning tools. And then like we go into in vivo in various animal models, looking at both pre and after vaccination to understand if specific cellular targets, let's say within the B cell compartment or innate immunity or like cytokines, could actually predict the antibody response or the pharmacological or PK/PD profile of these vaccines. And so we are employing this strategy in our clinical trials too. Hopefully we can then stratify patients based upon actual biomarkers that could be implemented in the future of this field, even against other non-opioid targets. As far as like other parallel programs, we are also working on antibodies. Antibodies have the advantage compared to vaccines that they're already made and they're active immediately. You don't need like to build up over months like the immunity. And so in this case, like again, like we start from the early discovery phase all the way like to IND-enabling studies. Our most advanced like program, we have a humanized monoclonal antibody against fentanyl and fentanyl analogs, for which we have completed all the preclinical efficacy, selectivity in mice, rats, pigs, and non-human primates. And we're in the midst of manufacturing, and we have planned like GLP toxicologies at studies at CROs, and hopefully we can start the clinical trial by the end of the year. Other pipelines, like we're developing antibodies against like a variety of other opioids, like heroin, oxycodone, carfentanil, xylazine, nitazine, etc. And also we are trying like to come up with like new ways to make antibodies or to deploy these antibodies using a combination of protein engineering, machine learning, and so forth. This is our platform. So we develop different workflows for high throughput screening of antibodies. These are probably common, like, you know, if you have a program in infectious disease like HIV and so forth, these are very common things. It's just that we have to apply it to substance use disorders and small molecule targets. We always start by isolating B cells, whether it's a mouse, a human, or like other species, and then from there, we either combine hybridoma technology with bulk sequencing or sorting with like sequencing of antigen-specific B cells. But bottom line, through these different workflows, we can isolate antibody-binding regions that then can be further engineered into an actual antibody that can be then processed or scaled up and so forth. As far as like antibody-binding strategies, we are using, I guess, like biotech standard. We work both on the Fab, so the binding region of the antibody, as well as like the FC portion. And the goal is like to make better humanized antibodies or understanding their biophysical properties and see if by using a combination of structural biology and in silico design, we can make like better, you'll say, higher affinity and so forth. Now I'm going to show you a couple of examples of how we do this. But first, just to give you an idea of how these antibodies can work, these are studies where in rats, we first like give enough fentanyl to receive, to achieve like an endpoint. In this case was like antinociception or respiratory depression. And then we reverse with either a MAB or naloxone as control. And as you can see from these panels like A and B, the monoclonal antibody is comparatively effective to naloxone in reversing fentanyl-induced effects. And when you're comparing the antibody to either IV or sub-Q, naloxone is somewhat in between, and this is where some of the earlier generation of like monoclonal antibodies. Sort of fast forward, some of the, I guess, like more improved like design like antibodies, like here we are scaling up and testing in mini-pigs. In mini-pigs, we have developed a model of apnea. So we can infuse fentanyl until the animals stop breathing, and then after 2 minutes of apnea, we reverse it with MAB or naloxone as a control. And what we're seeing is that the reversal of respiratory depression is equivalent to naloxone, and we confirm that like in panel B with the pharmacokinetics, like showing that as soon as you give in the MAB, there is that kind of shoot up in like bound antibody, bound like drug by the antibody. So once it's bound, the antibody-drug complex is inactive. So on the left, we are showing how, like, using relatively high, like fentanyl plasma concentration, where actually the antibody prevents activation of the mu-opioid receptor. So if in theory, if there are peripheral effects, those would be ablated. But also the antibody doesn't have any effect of its own, so there are less concern for side effects. And also by structural biology, we are seeing like how the antibody pocket is fully sort of engulfed in fentanyl. So essentially, if you take a unbound or bound like antibody, you don't see any structural differences. Well, instead you would see them, let's say, when the antibody would be bound to a virus. And in fact, to that point, like when antibodies bind things like viruses, or we've seen that a lot with SARS-CoV-2, the antibody will trigger then like other effector functions like NK cells, etc. We don't find any evidence of that with antibodies against like small molecules such as fentanyl, oxy, and so forth. Moving sort of forward, we kept like scaling up like the material for doing like for clinical trials, but also to allow larger animal models. These are non-human primates, on which like we developed this model of like doing dose responsive to induce like respiratory depression, and essentially reversing those like with antibody and comparing them like to naloxone. And so acute delivery of monoclonal would reverse respiratory depressive effect of fentanyl in monkeys, but also if each week we re-challenge animals like with fentanyl up to like 3 to 4 weeks, depending upon the individual, you would have like protection. So that would model like re-challenge, re-exposure, like in sort of repeated overdose scenarios. Going out more chronically, self-administration of fentanyl is blunted or reduced by antibody treatment for up to like 4 weeks. And that is selective because like cocaine and heroin self-admin is not affected. So we are hoping that MAB like beyond OUD can also, beyond like overdose, could also be used as a treatment for opioid use disorders. As far as like activity and selectivity, we always look at the binding profile of like all a generation of antibodies, and so we are showing that they have affinity for the target, but not for off-target compounds of interest. And then, as I was mentioning, like using antibody engineering coupled with structure-guided activities, we can actually really tailor these antibodies for their indications. So, for example, on the top, we have an antibody that binds both fentanyl and carfentanil, but on the bottom, we modified so that binds exclusively carfentanil and leaves like activity to fentanyl intact. Another example that to allow like potential treatment and probably feasibility in the clinic, we have inserted mutations in the Fc portion of the antibodies that could actually increase the half-life of these antibodies compared to the parent wild type. And so it can go as high as like 4 times like longer activity. So in theory, you could have an antibody that could be given every 3 months, 4 months, if that's the case. And also by using a lot of like these in silico tools, you can really come up like with new mutation and seeing like properties of these antibodies. Finally, like with always the idea of making something that is more scalable or is maybe like cheaper or more feasible or more amenable to storage, we also start working with like smaller like fragments or single domain antibodies that compared to a monoclonal antibody can be delivered potentially IM or IN, or could be stabilized like to be stored as a powder, and of course like provide like more intellectual property to actually advance and attract like investment to bring this like to the commercial space. And then in closure, like we are hoping that antibody-based strategies like would provide probably wider access or like more diversified portfolio of medications, like for the patients, for the physicians, for various scenarios. And then again, like this approach is selective and can be combined with current medications. So we see the value of combining, for instance, a heroin vaccine and methadone, a fentanyl MAB and naloxone to provide that kind of overall protection. And then like finally, I'm missing one slide here. So there was one slide, but you know, essentially now because of also the theme of this particular conference, like the idea is that we are trying to bring like some of these more advanced candidates like to the commercial space, and therefore we have now like an actual biotech company to bring this forward. And this is our team. Like we have our team in Washington and then at Minnesota, where I was for the past 20 years before moving to Seattle. And then we have our consortium for medication development that also include like either industry partners, etc. So thank you very much for your time.
Would people go into precipitated withdrawal? And will people go into precipitated withdrawal if you apply the antibody? And do you need to reapply how often?
That's a very good question. Like so far, like in animal models, we haven't seen any evidence of withdrawal, whether you do like somatic signs or you look by intracranial self-stimulation. We haven't seen it, and always we compare like to naloxone. So that's one. Also in our clinical trial of the oxycodone vaccine, like we don't see any withdrawal. Obviously like builds over time, so the likelihood is less. And then like as far as like giving the mAb, for now like the regular mAb is like once a month. The mutated mAb with a life extensive mutation could be like every 3 months, but now in the clinic in the infectious disease space, some of those mutations show efficacy up to 5 months. So that would be best wishful thinking, but I would say like 4 months would be our target. But again, it needs to be demonstrated like in actual humans. So thank you for your questions.
Organized by International Society of Addiction Medicine, sponsors and co-organizers, Scenray, Indivir, Spark Biomedical, Salofa, Ethifarm, University of Minnesota, European Union Drugs Agency, UK Office for Life Sciences,